I'd like to remind everyone that this meeting will be recorded and this will be made available on YouTube a bit after the webinar happens. We still need to process the captions and everything. Um, but this will be made available in addition to the demos we're going to show. Just to introduce myself, my name is Bill. I'm the lead for the GDC User Services team, and today I'd like to give you an overview of the GDC API. Just to give a quick agenda for what we're going to talk about today, we're going to give an introduction as well as a primer on general API usage. Some of this may be familiar as we did um, about 2 years ago, we did a webinar on the GDC API. This one's going to be more focused on some of the analysis endpoints, but if you'd like to know a lot of the basics, I would recommend checking out the one from, I believe it was April 2021. Um, so in addition to the general API usage, we're then going to go into the what most of the presentation is going to be, which is the API demonstration with a Jupyter Notebook. This is going to go over some basic API usage as well as some examples from the analysis endpoints. Um, During the webinar, if you have any questions, we would recommend you type the questions into the Q&A field. We'll answer them at the end. And I just want to bring to your attention that in order to get to the Q&A field, there might be these three dots here in the bottom right. You might need to click that to get to Q&A. There's another field called chat. We may not see the questions that come into that, so try to put your questions in Q&A. So to get started on the API, the GDC API, it's the API that allows data to be queried, submitted, or downloaded from the GDC. Um, in addition to the GDC, the API can be used by just about anyone um, because it is open and a lot of it does not require any sort of authorization. So the GDC API is also used by third-party applications. And if you look on the GDC website, there's a community tools section. You can see that many other apps do use the GDC API. Um, the GDC API can be accessed directly for anyone who knows how to use it, and there's a multitude of endpoints that can be used. These endpoints can be accessed via the command line using curl usually, the web browser, which is used in the same way as the command line, but a bit more static, and then also through Python scripts, which is what we're mostly going to be demonstrating today. So most of what you see in the GDC, if you go use the portal, if you use the data transfer tool, if you use any of GDC's features, you are using the API. Everything that the GDC does is built on top of the API. So if you see something done in the portal, you you can be fairly certain that it can be done via the API if you want to do it programmatically or if you have some ways that you'd like to customize it. The one exception I can give you is that tokens must be downloaded from the portal. They can't be automated because there's a special authorization system it has to go through. So everything except for token download can usually be done through the API. So just to talk about the API usage, and I'm going to go through this kind of quick because I want to get to most of the demos, and I think there's some webinars that have this information. But in general, the way it works is we have our GDC API main URL, which is api.gdc.cancer.gov, and then that's followed by a slash, and then the specific endpoint that you'll be using. After that comes the parameters that give information on how you want to query that endpoint. And we're going to get much deeper into this later. But just to show you the query on like a command line or browser, it's question mark after the endpoint, then parameter 1, ampersand, parameter 2, etc. In Python, this is actually laid out more like a dictionary data type. So you have a set of parameters and the and values, like a key value structure. And we're going to see that in a little bit. But Now to go over the specific endpoints that the GDC makes available. Um, the endpoints access information about whatever type they are. So the files endpoint gives you information about files, projects about projects, etc. So if you wanted to say a list of all files associated with a particular case, then you would use the files endpoint because each entry in the files endpoint is a file. If you wanted to say how many cases are associated with this project, you wouldn't use the project endpoint, you would use the case endpoint because each of the entries in that endpoint would be one case, so it could be parsed accordingly. Um, so the files, project, and cases are most of what you see in the section of the GDC where you download files, the repository. But in the exploration side, you'll see some more information that comes from the API, like mutations, genes, copy number variations. And these also come from different endpoints. The SSMs, the simple somatic mutations, occurrences, genes, CNVs. Um, we're going to show some examples for those later. Then there are some special endpoints called the analysis endpoints that do very specific things. So, for example, the top mutated cases by gene will show which cases have the most mutations for a specific gene, or top mutated gene by project in within a project, which genes are the top mutated. Um, so these are all designed specifically to show things like visualizations on the portal and also to provide specific analyses that users might want to do and may not want to write their own scripts to do. In addition to all these mutation counts and things like that, we have a survival endpoint that automatically generates survival plots. You may have seen the survival plots that are in the GDC data portal. The API can also be used to generate those, and a bit more customization is available through the API since we're touching it directly. Lastly, which kind of doesn't fit into either of these categories, but I stuck it into the analysis endpoints, is the BAM slicing, which is we're not going to see that today because everything we're going to be doing is open access and the slicing requires a token. But this allows you to take only a section of a BAM file so that to save space basically. So you can take a specific chromosome, a specific gene, a specific region, all that is customizable. Now to get into the parameters, just to go over these real quick, and we're going to get into what they actually do in the demo. But the first one we have is filters, which describes a subset of the data. The fields, this shows which fields you want to show, which properties. So these are these can be specified. The size is the number of entries you want to display, just in case you only wanted the top X, or if perhaps your query was too big and you wanted to break it up, you would use that. The format, things can be exported in JSON or TSV format depending on your preference and the structure of the data. The pretty parameter makes things human readable, and for JSON, it gives it a better indentation. From starts at an entry that's not the first one, and then expand shows all fields for a specific entity. So, for example, all fields for demographic, for diagnosis, um, for exposures, things like that. So if you wanted all the information about a diagnosis and didn't want to type out all the fields, this is the one you would use. And I think we're not going to go into that one today, but in the previous webinar from April 2021, we get into that. So I'd recommend checking that out. So as an example query here, um, this would normally be on one line if you were looking at it on the command line or in a browser, but it's broken up here for readability. So starting off, we have the main API URL followed by the cases endpoint. And this means all the entries that you get out of this are going to be one case. So the first thing we're going to do is specify the fields. the submitter ID, and the primary site. This displays the human readable case ID, so it might be like a TCGA barcode or a different. I know many projects use barcodes, something specified by the project itself, and then also the primary site, and they're divided by a comma here. And the primary site will be where the primary tumor was found, um, followed by an ampersand, and then the next parameter size. Uh, Size equals 100. This displays the first 100 entries, and while that might seem weird if you just wanted to see the first 100 things that show up, this will come into play later. So I'll show you a bit about that. Format equals TSV. This displays it in TSV format, so it can be exported and viewed in something like Microsoft Excel, Google Sheets, any spreadsheet program that you have. And then pretty equals true displays in human readable format. Now, one thing you may have missed in that previous query is how do I get what I'm looking for? As in, if you give me the first 100 cases, that does nothing for me. And that's true. I'm now getting into the filters parameter. Um, and without this, all the GDC API calls would just return everything, or the first X number of things that it could, depending on what you specify. There's not many use cases for that. So the filters parameter is pretty important. So this allows for a subset of items to be retrieved from the API based on a set of criteria. So, for example, I'm looking for all of the RNA-seq BAM files from patients in which the primary site is brain. So, in this case on the right here, this is a JSON formatted filter. We have cases.primary_site is equal to brain. Files.data format is equal to BAM. This just says give me the BAM files or the alignments. And then files.experimental strategy is RNA-seq. We specify all of these, and because this is an AND operator, it will show us all of the files. In this case, we'd be sending it to the file endpoint that have all of these criteria. So to talk about the operators, most people who are using an API um, or watching a webinar about the API are probably familiar with many of these, like equals to, not equals to, less than, that kind of thing. Um, but there's a few here that might interest you, like for example, exclude if any. Um, this will this will exclude anything that you specify. So if perhaps there was a list of 100 criteria, but you wanted it to not show you one of them, this would be the way to do it. Or this would be less computationally intense. So this one would work better. And then for ands and ors, I showed you an example of the ands before. It's just if you want to combine operations, this is how you do them. So now let's get into the actual demonstration here. So let me go ahead and back out of this and pull up the Jupyter Notebook. So this Jupyter Notebook was designed to be distributable, and it has lots of helpful links and such for using this notebook standalone. So you don't have to watch this webinar recording, though you could. Um, Let me bring this up a little bit so we can see more stuff. And special thanks to Katie from my team for putting this format together. Um, and at the beginning here, you will see that there's lots of information. And I'm not going to go through all of this at the top just for time's sake, but what we can see here is that we're going to need to import some packages to use um, during this all of these demonstrations. So in order to access the GDC API from Python, you will need the requests package. So that a lot of times it comes with Python, sometimes it doesn't. So we'd recommend running pip install requests, and that'll usually do the trick. If that doesn't, you can usually Google around and figure it out. Um, then the JSON package, that I don't think I've ever seen it not come with Python, but But yeah, we'd recommend importing that because most everything that goes through the API is in JSON format in some way or another, and this will help you parse it out. The URL library, we're not going to use much in this demonstration, but if you are doing some URL encoding, this is very useful. But we're going to go ahead and run this and import all these libraries so things don't crash on us later. So going down, this is some very basic information about how to use the request package um, and how the format is laid out. So we're going to go down to the start of this demo here, which is basic API queries. And the first thing in just about every demo that we do is check to make sure that you're connected to the API. This is a good first step for troubleshooting because you want to make sure that your computer can connect to the API. And this, I'm going to go ahead and run this. And this will specify, is there something blocking it, like a security feature, or is your internet out, or is there a problem with the connection? So here we could see status equals okay. We're good with that. And what I'm doing here is, and most of these demos are going to be structured this way, in that we have the main URL. We'll just concatenate the endpoint to it. So this is the status endpoint. We concatenate them. We run a GET request, so request.get to the endpoint, and then we print the URL to show what we're querying, and then the request text that comes out of that. So in this case, it's just kind of raw JSON. We're going to parse that a little bit later. Um, but just to show you that this works, we're going to take this status endpoint. And if you wanted to use this same call um, on a browser, you can go ahead and just paste that in there, and you'll see the same exact text here. So uh, any way that you'd want to access the API is your choice. So let's talk next about the cases endpoint. Um, this right here is just a default query to the cases endpoint, and there's not many parameters we're going to use here. We're going to go directly to the cases endpoint, and as you'll see as I go down, I'm going to introduce some of these concepts less and less because I've previously looked at them. So for parameters, we're only going to pass one, and that's using the pretty parameter. We're going to set that to true. So because the the default format is JSON, it's going to show the output here in indented JSON in human readable format. So we're going to go ahead and run that. And as you can see, we have the first 10 cases, and they have what are the default fields here. So we have a set of sample IDs, we have the primary site, we have the diagnosis IDs, the updated date time, information like that. And one thing I do want to specify is the case ID is a UUID. This is assigned when the case is created in the GDC. And then there is what's called a submitter ID, which is the human readable barcode, which may or may not have some sort of significance in terms of its structure. Um, so anyways, I only passed one parameter and it gave me the first ten cases in the API. Um, that that doesn't mean anything to anyone because the first 10 they're kind of in an arbitrary order. So the next thing we're gonna do is demonstrate a few more parameters, and this is still going to be the arbitrary first 10, but we're going to show you some more features you can use. So of course we used pretty before. It displays the JSON output indented. Um, the fields, it specifies the fields that are to be displayed. So up here we just have a set of default ones. Down here we'll be able to specify which ones we want. The format specifies the desired output. JSON or TSV. Usually XML may or may not be supported, so I wouldn't recommend using that. And then the size specifies how many entries you would like to display. So the below query will first display the submitter ID and project ID fields. Fields equals submitter ID and project.project ID. Display in a TSV format, format equals TSV. Display the first 5 entries, size equal 5. And then the pretty parameter, it won't do much here because we aren't using JSON, but the TSV will be human readable, so that will be okay. So what we do is we take this Python dictionary here, um, and we pass it to the request.get command using params equals and then the name of the field, which is also params. So we're going to go ahead and run this, and it should do what we set up here. We have the submitter ID over here, submitter ID, the project ID, and then the ID. And even though we didn't specify for it to display the case ID, the endpoint will always display the ID for whatever you're querying. So no matter what, the case ID will appear there. Um, so yeah, we have the first 5 here. And now to get into performing a useful query um, by implementing the filters parameter. Now, all the filters parameter really does is tells you to display a desired subset of entries. So this can be specified in JSON format, and the filter I'm going to specify below only will show cases from the TCGA-LUAD project. So that is specified with project ID or cases.project.project ID equals TCGA LUAD. So filters is specified here, and then when we pass it to the parameters, we have to use the JSON.dumps filters so that it shows it as a string, um, so that it can be read by the API. So we're going to go ahead and run basically the same exact command here, and as you can see, we're only showing the first 5 now, but we could have more if we wanted to see all of the cases in this project. And yeah, because we specified this project, all of the things we're looking at now belong to it. Um, so that is a simple filter, but you might want to do something a bit more complex. And we're going to demonstrate that using the AND operator that was shown in the presentation earlier. So the this is almost the same query as before. Almost all the same parameters are there, except for a few key things that I'm going to show you. One, the filters is changed to have an AND operator and have 2 separate operators included here. So we have the same one as before. Project is TCGA LUAD, and then we have um, a second um, a second filter that says the cases.demographic.gender. So the gender field is equal to male. And I just want to bring up here it's equal, and the operator encompassing both of them is and. So, and these are kind of structured like a list here. So we have the square brackets that surround them. Going down to the parameters, it's mostly the same, except I'm having it display the demographic.gender field so that we can display that this field worked. And we're also going to show the first 5 again, just because it's small and easy to look at. So here we have the first 5 cases. As you can see, they all belong to this project, and they are all have a demographic.gender as male. So a frequently asked question that we get is, how do we know the names of the fields? Because if you look up here, they're not always that intuitive. Like cases.project.project ID. How do I know to put cases.project on there? I might know project ID. And then for gender, cases.demographic.gender is the proper field. So we want to know, how do I know the names? And this is actually pretty easy. we have an endpoint called the underscore mapping endpoint. And what you do is you append underscore mapping after a slash to whatever endpoint you are querying. So for cases, you would query cases slash underscore mapping, and it will give you a JSON format that is not indented. So what you'll need to do is run just a real quick process or um, post-processing line here that loads it up into JSON and then indents it when you print it. And here you can see it kind of gives this long, complex JSON that specifies a lot of things that are mapped out in this endpoint. But what we are interested in is just the fields, which I won't scroll all the way through this, but there is a key in here that shows fields and then a list of names. So we're going to format this a little bit using the JSON package again. And what we're going to do is only display the fields key. So I'm going to go ahead and run that. And as you can see, we have a clean list. Whoops. We have a clean list of fields that can be used for the case endpoint. If you wanted to see a list of fields for the files endpoint, you would use files underscore mapping rather than cases underscore mapping. And that works for any other um, endpoint. So if you have any doubts about if your field is correct or not, this is a good way to check it out. And then also, I want to bring to your attention that if you have a list of fields and perhaps um, you're unsure if one of them is correct, if you format your output in JSON, which actually I'll demonstrate real quick. If you format your output in JSON, which I can do for this one up here. So do JSON, and then we'll do project ID, but we'll forget the D here. So I'll go ahead and run this again, and you'll see it gives me information, but it says at the bottom, warning, unrecognized value, project.project I. So this is a good way to troubleshoot an API call that you're making. So JSON format will give you all the information that you can possibly need. So going down to the next demonstration here, we want to talk about the analysis endpoints. And here with the analysis endpoints, I'm kind of lumping in everything that is on the portal exploration page in V1 and all of the special analysis endpoints. And I'm going to show you a few select examples of how this works. So for this one up here, we're going to query the SSM's endpoint. So for mutations, there are simple semantic mutations and copy number variations, CNVs. Both of these are present and available. Here, we're going to look at simple semantic mutations. CNVs will come later. So we have the SSM endpoint. We will add that. And for the filters here, I'm going to query the any case that is over the age of 50 and under the age of 60. So I can do that with the greater than or equal to 50 years old. So it's 18,262 days. And then 60 years old, which is um, 22,279 days. Um, so then we're also going to further query that using the cases.project.project ID and just show me the SSMs that come from the project TCGA GBM. So these are simple semantic mutations that come from patients between these ages and that fall into this project. Under fields, we're going to see the genomic DNA chains. We're going to see it in a JSON format. And then the size here, I'm going to tell you about that in a bit, but I set it to 20,000. So let's go ahead and run this. And one of the things you may be asking is, how do I know what to make the size? Because if I go under, it's not going to show me the complete data. And if I go over, it could time out. So one thing we're looking at here is the total. So no matter what you set your size to, it's always going to show you a total. So we know there's 14,548 entries in this list that meet these criteria. If we set it to 20,000, that's not too much over the total, and it is over, so it will give you the complete set of data. If you wanted to like really quickly figure this out, you can do a size of like 5, something really small, and it will still give you that same number for the total. So you can go back and then run it again with that information in mind. So you'll see that I set a lot of sizes for this particular notebook, and that's how I got most of them by looking at the total size. Um, so next we're going to use the genes endpoint. For this one, of course, every entry is a gene, and here we're going to use the same age range as before. We're going to use the same project as before, but we're going to use this field, gene chromosome is equal to 2. So the genes must be on chromosome 2 and then have mutations in patients who are this age. Um, Under fields, we have some pretty useful things here. Biotype is the type of gene it is. So is this a protein coding gene? Is it some sort of RNA? Is it something else? And then symbol is the human readable symbol that usually stands for something. So the regular ID for genes is the ensemble one, which is like ENSG and then some long number. Um, and then the symbol is the one that those who study these genes will understand. I do want to bring to your attention that not all of the symbols and IDs are a one-to-one mapping, so keep that in mind if you are making queries like this. So with this information, what we have here is, um, let's go to the top, and we have this JSON format here. We have the data. Under that, we have a key called hits that has a list as its value. And then we have a bunch of smaller JSONs that have the keys ID, biotype, and symbol. So what we might want to do is just pull the symbols out for all of these into a big list. Um, so what you can do here is do something like use data, which is the name of the full text. Uh, Show me what's under the data key, and then show me what's under the hits key, which is now a list, as you saw up here. And then you can loop through it and add the gene symbol to the gene list. So when we run this, we can see that we have this giant list of genes, and you may need this for an analysis or a figure or a supplementary table. So you can do whatever you want with the list here. You print it out. So it's much easier to deal with in a list format than the other one, but it's pretty easy to go back and forth, of course. Um, the SSM occurrences endpoint, I do want to bring this to your attention that SSMs, simple semantic mutations, and SSM occurrences are two different things. So a mutation on chromosome 3 at position 100 that goes from A to G is a simple somatic mutation. This is a mutation that could occur in many people. The above mutation happening in this particular case is an occurrence. So it's a simple semantic mutation happening in the context of a case. So in this case, what we're going to do is we're going to query for the occurrences that happen within the same age range that we were talking about before, within TCGA GBM, also chromosome 2. And this is basically the same query as before, but instead of querying for specific mutations, which would be associated with multiple cases each, we're querying mutation occurrences, each of which is associated with one case. So, as you can see here, in the fields, we wanted to specify the case submitter ID. So this is the case to which that mutation occurred in. So query of case submitter ID, and we have a giant list of lots of them. Um, so one thing you might be interested in is which of the cases has the most mutations, which in this instance would be which of the cases appears the greatest number of times. So here I'm using this other package that came with my version of Python called counter. Um, and I'm just making a giant list of all of the case instances and running a counter on it so that it displays each case with the number of occurrences. So in this instance here, we have this first case having 537 mutations, and then it gets a lot lower after that. So this could be of interest to somebody who someone might want to look into this case and say, okay, is there some batch effect? Is there a reason why this case has many mutations more than the other ones? Um, or it might be interesting to see which cases have fewer um, simple semantic mutations. So this is just an example of some post-processing that can happen here. Um, the CNV endpoint shows the copy number variation. Um, In TCGA, the CNV data are based on the ASCAT2 pipeline, which is run using the Affymetrix SNP6 data. So this is the data that you'll find in the API. Here is just some CNVs that can be observed in chromosome 2 in the GBM project. But I'm going to skip over that real quick to show the CNV endpoint as it can be used for a single case. So here, what I'm doing is querying all of the copy number variations that can be observed in this particular patient here. So I'm going to run that because I think this one might take a while. But what we're going to display is the NCBI build, the chromosome in which the CNV is found, the gene symbol for which the gene in which the CNV is found, and also the CNV change. Is it a gain or a loss? So once I run that, you can see that I have a list of CNV changes, chromosomes, and genes. And this actually might be more useful as a TSV, so I'm going to go ahead and run that. And for something like this, um, you will see that we have a nice list of CNV gains and losses for particular genes um, on particular chromosomes. So this could be parsed out in Excel or Google Sheets or just as like a TSV in Bash. So next I want to talk about one of the analysis endpoints called the top mutated genes by project. And thank you to Himanzo for putting together this example. And this will show users the top N mutated genes within a particular project. So the endpoint here, analysis slash top mutated genes by project. And here we have the program, which is TCGA, and the project ID, which is TCGA GBM. So we're going to go ahead and run this. But as you can see, the size parameter will tell you what your N should be. So in this case, it's going to be the top 100 mutated genes within a project. So here we have the top one, 456, all the way down to the 100th one, which wouldn't be worth it to scroll down. But the size can be changed to make it the top however many you're looking for. Lastly, I want to talk a little bit about the survival analysis. This is the endpoint that can be used to show the survival Kaplan-Meier curves and can compare different sets to show um, how different or how show whether or not they're significantly different using a Kaplan-Meier estimate. So we're going to go ahead and this one is separated out with each set being an item in a list. So the first item in the list is just TCGA Brica, this project. The next one is the TCGA SKCM. And the third set is TCGA LUAD. So these are what we're doing is we're taking the cases for these three different projects. We're comparing them. We're comparing their survival values using a Kaplan-Meier estimate, and then this will generate a survival curve for all of them. And using the time and the survival estimate, if you plug this into like a graph program, this would generate the actual curve. Um, but at the bottom here is more information about the overall stats, comparing the three cohorts. And as you can see, the chi-squared value is pretty high and the p-value is very low. It would be no surprise that these three cohorts have significantly different survival estimates because, of course, these are three different cancers. I don't think this tells us much, but it's a demo for how this survival curve feature works. For a slightly more useful one, we're going to try to look at TCGA survival by BRCA subtype. Um, in this case, the cases are passed as a list of submitter IDs, and each list is divided into subtypes using a modified supplemental file from the 2012 TCGA BRCA PanCan paper, which is a link here. This will lead to the GDC publication page for this. I'm going to run this because I think this one actually doesn't take very long, but um, just to show you a little bit about what we did, here I have this file here called subtypes.txt. This is actually um, modified from a TSV that's available on the publication page, and we just have a list of barcodes and the uh, the subtype that was determined here. So some are basal, some are luminal, and some are HER2 subtypes. So here we have these lists here, luminal A, luminal B, HER2, and basal. And what we can do here is use different combinations of lists here. HER2 is the list of case IDs that have this subtype, and same with luminal A. And we can do something like check out uh, luminal B, compare with luminal A, and run this query. Now, as you can see, what we're doing is we're pulling out the overall stats. If you didn't do that, you can look at all of the values for the curve and export that. But here we're just looking at the stats. So here we have a pretty low p-value for something like basal and luminal A. You can compare those two and see that they have a slightly higher p-value, well, a much higher p-value. Um, so anyways, that is the end of the demo. And let me just show you a few more things and then we can get to questions. Um, so I just want to go over a few GDC announcements. Um, so we have an upcoming data release 37, which will be in March of 2023. In this release, the remainder of the legacy files that will be migrated to the active portal will be migrated there, as well as the release of several new projects, which include the Apollo Luad project, the CGCI HTMCP lung cancer project, and then two projects from the Match program, ARMQ and ARMY. Then we also have some new datasets from TCGA, Target, HCMI, and CPTAC3, which you'll see more information on in our release notes. The last thing I want to mention is that the legacy portal retirement will be happening in the release after data release 37. So this is likely to be April 2023. So please, if you need any files from the legacy archive, start downloading them now. They will no longer be available after retirement. If you have any questions about any files or anything in the legacy archive, feel free to reach out to the help desk and we can make sure that we can either assure you that they will be migrated somewhere else, maybe to a publication page, or we would advise that you download them as soon as possible. So just a bit of a warning there. And lastly, if we have any questions from participants, we can take those. But just to show you some useful links, we have our documentation site, which can be used to look up information on just about anything I showed you here. We have our website and the GDC help desk. I recommend contacting them if you have any questions about this webinar, the Legacy Archive, anything else that goes directly to my team. And we'll answer your question. We'll respond to you within a business day. Some questions take longer to do that. But anyways, that's it for the presentation. Are there any questions?
Hey, hi, Bill. So we have about 9 questions. I'll go through
Oh,
the
wow.
first one. Does the R API have the same functionality as the Python one?
Um, yeah, so I'm not too familiar with accessing the API via R. I know it can be done, and every every endpoint that can be accessed via Python can be accessed via R. Now, I can't tell you what the post-processing is going to look like there. I'm sure most of the same functionalities are available, but yeah, in general, anything you can do with Python, you can probably do with R. It might take some extra work. And I also want to mention that I know there are some GDC like some GDC specific packages that can be run via R. Some of those have special things in them that, like pulling metadata from other sources. The GDC really has no control over that, so I can't really speak to how those work. But in general, yes, R can do anything with the API that Python can.
Yeah, I was just going to add to that was it's a REST API, so it's language agnostic. So as long as you can use REST from whichever language, you can pull it and create a query. So the second question is there any size limitation for the API command, such as size is equal to 2,500?
Yes, I you know, I should have researched this before this webinar, but I'm pretty sure it's like 20,000 for the files and the cases, and then something higher for the SSMs. I can't remember exactly what they were, but it wouldn't be too much of a hassle if you were to try it like on your own. Because if you try something like a million, you'll see when you press enter, it'll just kind of like do nothing. But if you so yeah, if you were curious, you could work that out just by kind of you know doing some min-max tests, and that should give you the information you need.
And we prefer you to throttle it so that it helps us. Okay, third question. How do we get the list of all endpoints, parameters, values given parameter and endpoint so we know what to filter on?
Yeah, good question. So for the things you can filter on, you would go to the mapping endpoint, like I demonstrated before. But for all of the endpoints, I don't think you can access them anywhere specific. So I'd recommend looking at the API documentation or this webinar. I believe we have most of them laid out there, at least the ones that you would need to use.
Yeah, I would say the best source is the API endpoint where you have the API docs. They list them. Yeah, we don't have a Swagger that will give you the endpoints yet right now. For cases endpoint, but they're all listed in the documentation site. For cases endpoint, are the IDs unique? Are the IDs unique patients? And what if a given patient have multiple samples taken at different time, for example?
Yeah, that's a good question. So for the cases endpoint, the each of the IDs are a specific patient. So within that patient, you could have multiple samples. So if you wanted to get that information from the cases endpoint, um, that I would recommend doing in like a JSON format because you could have nested lists with like many to many relationships. Um, However, if you wanted to look at like, you have a list of files and you know that these files came from different samples from the same case. So what you could do is use the files endpoint, query for the cases dot samples is what it would be, and that information would map one to most files. So you would have a list of samples, and then you could also have a list of which case that sample maps back to. So um, you would have a list of files. Each file would have a sample, and then each sample would have a the case it's associated with, and that would be a good way to show kind of a many to many relationship in a very flat TSV way. And that's, to my understanding, one of the most common ways users will gather information about GDC files.
So just to add to that, Bill, so what you can do is you can have both fields and filters. So the fields lets you narrow what data you want back. So you can put a case ID, a sample, you can put samples, cases as your field, and then you can do a filter based on a particular sample or whatever you filter as a disease type, and it will give you back the samples associated with them. So that's another way of doing that, what Bill just described. So look up fields. Then the next question. GDC data model is really impressive to store cancer data sets project. I am wondering if there if there are any similar multi-omics projects for immunology diseases from NIH.
Ooh, I'm not sure about that one. Himansu, do you have any ideas?
No, I don't. Let me put Peggy on. If Peggy's on, maybe she knows. Or if anybody else knows, I can get to. If anybody else knows, put it in the chat. Well, I'll reply back.
All right, let's see. The next question,
Okay.
are we able
Next
to retrieve?
question is there any demo code for this tutorial based on R?
Um, Not for this particular one, but I know there are R packages out there that specifically query the GDC API, and I'm sure they do include demo code. Usually, there will be something like that there. So, if you look at just
BioLinks? Yeah.
yeah, TCJ BioLinks is one of them. I know there are other ones. So if you just query like GDC under R packages, you usually find something.
Are we able to retrieve TA dinucleotide repeats from the current GDC database?
So I don't think that information is specifically stored in the API. If somebody tested for it, it might show up under molecular tests, but I don't think we have that specifically at this time. So that would be something you would have to query around the actual, variant calls and the BAM files if you had access to those.
Okay. Are we able to retrieve drug information for the cases?
Yes. So it depends whether or not drug information is populated for the cases. If they are, they'll usually be found under the fields with the word treatment in it. So if you were to query the mapping endpoint and you were to look for the keyword treatment, that will be where you find the drug information. In addition to that, if not using the API, there are a bunch of um, clinical supplements, and if the information doesn't make it into the API, it'll be in the clinical supplements.
Yeah, and you can also create a filter on a particular drug if you want to. You can check the GDC dictionary for the drugs that GDC supports. So, um, if you wanted to return a list of cases from primary site, brain, disease type gliomas, and wanted to match on a specific gene symbol such as IDH1 or EGFR, would you use cases, follow up, molecular test, gene symbol for assembling your filter query?
Yeah, that's a good question. So that particular query there specifies the gene symbol that is tested in clinical data. So this would be a clinical test that is used. The SSM's endpoint that I was using before is done using the bioinformatics files in the GDC. So this would be the whole exome sequencing variant calls. So if there was an IDH1 or EGFR clinical test that was done, yes, that would be the symbol to query. However, if you want to know if the variant calls found mutations within those two genes, you would use the SSM's endpoint. And I forget exactly what the gene symbols um, what exactly the gene symbols I guess field is, but so that would be kind of two different tests, whether it appears in the clinical data or in the molecular data.
Yeah, the difference is the tests are what is provided by the submitters as part of the project submission, whereas what's the in the analysis note, the SSM endpoint is the analysis that was performed on the data. So, okay, see, there's one couple more. For cases endpoint, are the IDs unique patients? What if given patients have multiple samples? I think we answered that one, right?
Oh yeah, I think we already did that. So
Okay, so one
I do.
more, I think. Um, so I think the slides would be shared. I think the slides will be shared after the meeting, correct?
Yeah, so we won't be sharing the slides directly, but we will be sharing the this as a YouTube video, as well as the Jupyter Notebook will be made available through GitHub. So you'll be able to see it there.
Okay. Any other questions?
I did find another question that I think we skipped. Hold on, let me make sure I can find it. I want to know if we were able to get the information about an updated version of a specific file with the API. And the answer to that question is yes. We didn't demo it in this specific webinar, but there is an endpoint called the history endpoint. And what this does is it tells you which file is the latest version, and if there are previous files, it'll provide the UUIDs for those. Usually, those files can still be downloaded if you were to um, directly query them using the API or the data transfer tool. However, I do want to say that it's not guaranteed that the previous BAM files in the previous slide images, of which I don't think we have any previous versions of those, but there's no guarantees that those will be stored simply because they are very, very large. So if you need any larger BAM files that are previous versions, I'd recommend downloading them now. If they are just something small like gene expression, for example, from HT-seq, which is no longer on the portal but can still be downloaded, those will all be available in the foreseeable future since they're very, very small.
So, Phil, there was a couple other questions. Is the Jupyter Notebook available somewhere? And also, there was a suggestion, could you also share the PowerPoint in the same GitHub folder of the Jupyter Notebook folder?
Yeah, so we'll run this by GDC leadership. We'd most likely share it as a PDF simply because those can't be modified too easily. Um, but in um, sorry, I believe there was two. What was the first question? Sorry.
The Jupyter Notebook be available? So,
Yes, the Jupyter Notebook, the exact one that I show you here, will be made available.
yeah, and we'll look into the PowerPoint being made available as a PDF. Oh, so I think that's about it. Um,
Awesome. Well, yeah, it looks like we're at time. So thank you everyone so much for joining, and I hope you all have a good rest of your day. See you later.
Thank you, everyone.